So I want to thank Mark very much and the other organizers for inviting me to speak with you today about the development of a system and looking at available systems to ask questions of what are things that we think may be contributing to leukemogenesis in children. Can we test that in a relevant biological system to assess whether and how and potentially how to intervene? I want to begin by making some remarks about how I ended up getting into this area. So I have, since beginning my postdoctoral work at the University of California, I've been very interested in how genetic changes, when it combine, affect cellular growth and lead to development of leukemia. And a number of years ago, I was presented with the possibility of basically it was a request for applications from the Electric Power Research Institute. Now, the reason that the Electric Power Research Institute, or EPRI, is interested in leukemia models is that is some epidemiologic evidence saying that under some circumstances there is an apparent association with very high levels of EMF exposure potentially being associated with risk of leukemia. And there's been some issues of biophysical implausibility. There have been some ideas about maybe it's not the EMF exposure itself, but other aspects of the electrical grid. And this is really an unproven area. But what EPRI looked at is they said, there really aren't at the time very good models of animal models that are representative of pediatric acute lymphoblastic leukemia that would be well suited to studying questions about environmental causality. And they funded work to develop such a model. And that's what I have worked on. And I'll tell you about the model that we have worked on and developed. And that we are just beginning to apply to answer these kinds of questions. I do note in the disclosure that one thing that's been great about working with EPRI is that I've been able to work very independently, and we're setting up our EMF studies that are just beginning in a way that myself and no one in my lab will know which are the exposed groups and which are the unexposed groups, which can be an important aspect in evaluating potentially controversial environmental exposures in regard to disease risk. And then what's been really wonderful for me as a scientist as well has been to be introduced to a new area, and through the development of this model, I've had the opportunity to participate in a number of novel research activities that are funded through the National Institutes of Health and now recently through US EPA. So the goals of the research were initially to develop an animal model of pediatric lymphoblastic leukemia, to utilize the model to assess the impact of environmental exposures on leukemogenesis. One of the things we've been very interested in is, could we establish some early endpoints? Leukemia can take, in children, it can take years to develop. In mice, it also can take a substantial period of time. And might we get some clues about effects or mechanisms of effect by looking at some early endpoints, some early measurements? And one theme of this meeting has been the importance of interactions and assessing not only the impact of environmental exposures in relationship to genetically modified models of pediatric leukemia, but also looking at issues of how do the exposures relate to stage of development and relate to alterations in the immune system. And one thing about working with an animal model is we have the possibility of varying the genetics, varying the characteristics of the immune system, varying the exposures, doing combinations of exposures in a way that can give us direct experimental insight into interactions. So these are the three parts that I'll talk about. The first two have come up, and I'll move relatively quickly through these areas. So these are pictures of 4 healthy children, and that sort of the issue really is that healthy children can go on and be impacted by a diagnosis of lymphoblastic leukemia. Now, after about 65 years of efforts and of progressive success, about 90% long-term survival for childhood BALL. But this entails 2 to 3 years of treatment. There's intensive induction chemotherapy, then ongoing consolidation chemotherapy, maintenance chemotherapy. This is a very significant impact immediately on the individual and on the family. And then ultimately, there are emotional, neurocognitive, and fertility effects. There also may be some effects leading to increased risk of second cancers. So a question that parents have often is, why did my child develop leukemia? And so we're trying to understand ALL development. So what are things that are suspected of being contributing factors? Well, inheritance of risk alleles from parents. So genetics can mean a number of things. One thing can be that we, of course, all inherit genetics from our parents, and that some of us and some children inherit from their parents changes in genes that might make them more susceptible to development of leukemia. It could be more susceptible to the effect of an environmental exposure. It may be more susceptible to developing cells that go on to develop additional genetic changes. There's also the acquisition of genetic changes that occur during life. And we know for childhood leukemia, for certain subtypes, in fact, the major subtypes of early childhood leukemia, that there are changes genetically that happen in the child before they're born that are associated with the development of leukemia. And then changes can happen postnatally as well. And then, of course, there are issues of environmental exposures. And this can be preconception. It can be, for example, effects, potentially there could be effects on sperm development that may be, confer a risk of disease later in life, and can be post-conception in the mother, prenatal and postnatal in the child. There's also important issues related to the immune system and the way that the intrinsic development of the immune system can matter for the development of cancer, including leukemia, but also the way that the immune system interacts with infection. And there's some really interesting findings in regard to childhood leukemia, for example. In certain populations, sending a child to daycare very early in life seems to confer a protection against the development of leukemia. Whereas there may be certain types of infections that may occur later in life that might increase risk of leukemia. And all these issues and the issues that come up here in terms of late exposure to infections, we know ionizing radiation is a well-established risk factor for the development of leukemia. A number of these other issues have already been addressed. Either Catherine brought them up in her talk, or they came up in other settings in this meeting outside relationship to leukemia, but that we have suspects and that we'd like to be able to understand in more detail whether they're contributing to ALL and what they're doing. So, of course, combination of inherited genetics, acquired mutations, infections, exposures, all together interact in the development of ALL. And the interaction is very likely to be important. And how do we sort it out? Well, one way we try to sort out the transition between, let's see if I can get it, between normal blood cell development. This is actually a picture of normal blood cells from a mouse going on to develop leukemia. Include epidemiologic approaches, and there's a lot of strength in the work that's being done, multivariate analysis, the availability of some prospective and population cohort studies. But being able to test the biology in model systems, testing the impact of variables can be quite useful. So, what are the animal models of ALL that are available? There are dozens. There are dozens of animal models of ALL. There are three main types, and a little bit of background on this. So one way of developing a model of leukemia is from an individual mouse or group of mice to take their bone marrow out, to genetically modify that bone marrow, and to put it back into mice. So this can be a very useful tool. It is one way of doing model development, but because of the variability that comes from modifying individual animals and sort of setting up one mouse at a time, this isn't a particularly favorable system for sort of a large exposure study. Xenografts refer to animal models in which human ALLs are placed into mice. And there's been a lot of recent success with this. It can be incredibly useful for testing ideas about therapeutics. But again, these are already established leukemias, so they're not ideal for looking at how do environmental factors influence leukemia development. And then the mainstay of looking at exposures is genetically modified mice. And there are modified mice that correspond to quite a few varied genetic changes of human ALL. So here is a pie chart that represents major different genetic subtypes of ALL. And some of you are going to be familiar with this type of a chart and information, but to others of you, sort of explaining what this shows is that human childhood ALL, it's not really one disease. It's a disease that has many, many different genetic changes and subtypes of genetic changes in it. And it's quite possible that certain effects, for example, might influence, let's say, the development of ETV6-RUNX1, which in Catherine's talk was referred to as TEL-AML1. It's an alternate name for the same gene. That maybe that if it something affects the development of that type of leukemia, but doesn't affect the development of another type of leukemia. And that's a major type of childhood ALL. And you can see there's a whole host of different types of leukemias. The major type of, there are two main types of lymphocytes, B lymphocytes that make antibodies and T lymphocytes that are involved in cell-cell interactions in the immune system. So if we take those out, this is a picture of the genetic subtypes of ALL. And for all of these that have stars on them, there are established or have recently been established models, mouse models of ALL. So there's we already are in a very complex field. We now layer onto it the fact that, well, which model should we test? Because any exposure that we want to test in any model is a quite substantial effort and money to be able to test a particular hypothesis. And so one of the things that we were very interested in was the two most common subtypes, which are this ETV6-RUNX1 and the hyperdiploid. To date, there is no good model of hyperdiploid ALL, whereas the ETV6-RUNX1, there are now some models available. Another very interesting feature of this subtype of ALL, this is a disease that starts before birth. This genetic abnormality is found at birth in the little bit of blood that's taken when children are born, even if they develop leukemia years later. So it's sort of a setup. A child may be born with this, and then depending on what happens to them, they may or may not go on to develop leukemia. There's some evidence that many more children are born with this genetic abnormality than actually develop disease, and that therefore there's the opportunity for the environment to be influencing who goes on to develop disease and who does not. So I'm going to talk a bit about animal models of leukemia and what they are, how we understand them, what we can do with them. So for each model, there's a particular genetic change that's been introduced. It's expressed in the mouse in a particular way. The enhancer promoter basically is like what cells are expressing it. It can be something that's turned on all the time, constitutive, or is something that you can turn on and off in a regulatable way, which can create some useful features. Some of these models involve mouse genes. Some involve human genes. And that choice can make a difference in terms of whether the mice develop disease and how they may respond to the environment. So there's this very interesting thing. Humans, we're almost all genetically different. There are people who are identical twins, but we're totally heterogeneous, and our exposures to the environment and how we respond to them are going to be dependent on our individuality. Most studies in mice are done in inbred strains. In other words, every mouse is like they're an identical twin. And the advantage of this is that you can you have a less noise in the system. You can say, well, they're all identical, and so they're all going to be responding the same way. The disadvantage is if you're in the, quote, wrong mouse strain, you might miss an effect that would happen in a different genetic background. And then there's a stimulus. Do you just watch and see whether the mice develop disease, or is there some interaction that you introduce to sort of stimulate disease in the system? So this basically breaks down to mutations, target cells, on or off. So I'm going to go through 6 models to give you a sense of the types of models that are out there and the work that's available. And this is really sort of to provide information about what is available and what types of things it's possible to do going forward and that we are beginning to undertake. So basically just normal inbred mice develop leukemia if you just follow them at a low percentage. So this is an older study from 1993, but up to 3% of a particular strain of mice, C57 black 6 mice, will eventually develop leukemia. So in theory, you could do studies just in mice that are not genetically modified. Another well-used model for understanding leukemia is a model that involves a gene called BCR-ABL. So this is a fusion of 2 different genes that have been brought together. It's involved in cell signaling. It's a type of genetic change that's present both in a disease called chronic myelogenous leukemia and in acute lymphoblastic leukemia. It occurs in children with ALL, but not that common. And it is the people who have this fusion are the benefits of a relatively recently developed therapy that can bind to this protein and essentially turn it off. So this model, there's a model in which it's basically expressed in most cells. It can be turned on and off. A human gene was used, particular mouse strain, and you can basically turn this gene off by most of the time having them on an antibiotic and then removing the antibiotic in a way that they've been genetically manipulated to then the gene will go off. And so here's a picture of even within this type of a model, these lines on the left are basically showing the survival of the mice of 4 different independent lines of these transgenic mice that were created, and all of them get sick, either faster or slower. And so this is one model that's available. Now, the fact that the mice get sick quite quickly, and all of them get sick quite quickly, means this is probably not a great model for looking at environmental modifiers. This is a very interesting model. It's a deletion of a gene called PAX5. So a very important type of protein in cells is what's called a transcription factor. It is a protein that regulates what other genes and proteins are expressed in cells. And that the decrease in the PAX5 transcription factor is seen in about 1/3 of children with ALL. Now, interestingly, these mice, if you just remove one copy of PAX5, they're basically fine unless something else happens to them. So in this model, basically these mice were treated, they wanted them to develop the common type of human childhood leukemia, B cell type of leukemia. So their T cell organ, the thymus, was removed, and then they were treated with 2 different types of carcinogens, a viral carcinogen and ENU. And so these mice do go on and develop disease after exposure to ENU or this viral carcinogen. It's an endogenous virus in the mice that will lead to leukemia. So this is one model, and it is a model that can be used. There's some really interesting recent data in which someone took this model and they basically, the story here is one of these interesting stories in science. So often we keep mice in a very clean place. We make sure that they don't get exposed to any infections because we want to have them not get sick from mouse illnesses. So what happened was one lab had these mice and they were watching them, they weren't getting sick and they decided, okay, we're going to save money. And we're going to take them from the super clean, non-infected place and we'll put them somewhere else. And when we put them somewhere else where there were infections among the mice there, the mice developed leukemia. So that basically they were in a place without pathogens. And then after a period of time, they put them in a place where there were pathogens. Only after the fact did they figure out what pathogens were there. There were viruses, bacteria, and parasites that were all that the mice got exposed to. And that on the left, what's showing here is that they didn't get sick at all if they didn't have any infections. but if they had infections, a subset developed illness. And this is a potentially very interesting model for looking at the interactions of environmental exposures, potentially interactions of environmental exposures with timing of infection and during the life of the animal. The ETV6-RUNX1 is representative of a change that's seen in about 25% of human childhood BALL. It can be expressed in various blood cells, and there's a recent model that's been described in which a small number of the mice went on to develop leukemia. Now, there have been lots of this type of model, this ETV6-RUNX1 model that were developed, and basically in almost all the models, the mice didn't develop leukemia. Now, maybe if they had all been put into a place where there were infections, they would have developed leukemia, but most of these were done in facilities where there were no infections, and the mice did not develop leukemia. So the work that my lab has worked on has been making use of the ETV6-RUNX1 as an introduced change, a transgene, expressing it in blood cells and in blood vessels. It's an inducible system. In other words, basically, it's not necessarily on initially all the time, but we can turn it on in the cells of, it gets turned on in the cells of interest. And what we found when we looked at these mice is they did not get leukemia above background. They were like wild-type mice. But when we introduced a known cooperating genetic change, the mice go on and develop leukemia. And I'll sort of show you a little bit more of the data. So this is sort of a picture of the transgene that was used. It's an inducible transgene, and this is a technology that enables basically, this piece basically says the gene is off, and then by making use of a protein called CRE, you can turn the gene on and get it expressed. And you'll notice here that it's not only the gene itself, but there's a downstream gene that says EGFP. This stands for enhanced green fluorescence protein. So when laser light is shown onto cells that have this protein, they turn fluorescent green. It's actually a protein that was originally identified in a fluorescent jellyfish. So we cross this into the CDKN2A background. The cells can be tracked with fluorescence. The transgene does have some impact on lymphocyte development. In fact, it's something that's associated with B-cell leukemia, but what it actually does initially is it suppresses the development of B cells, so that it almost seems counterintuitive. You'd think something that promotes cancer would tend to promote the growth of the cells that causes cancer, but it's in fact initially the reverse. These mice develop some BALL. The CDKN2A knockout mice develop BALL, even in the absence of tel-AML1, but we were able to see a combinatorial effect. So what I'm showing you here is a chart from a fluorescence activated cell sorter, and what you're looking at here is this represents light that's been scattered in a way that tells you how complex the cells are. Are the cells pretty simple inside, or do they have a lot of internal complexity in them, like granules, which a particular type of cell called a neutrophil has, whereas lymphocytes don't have much complexity and they show up down here. The GFP is basically, are they fluorescing? And what I want to show you here is this is a related mouse where you can see there's some fluorescent cells and some non-fluorescent cells. And then when we have our mice with tel-AML1 or ETV6-RUNX1, there's actually relatively few of the cells that express the green fluorescence protein, both in the lymphocytes and in the neutrophilic cells. So this is an interesting observation that there's that the transgene impaired growth of the blood cells, and we could follow that. Now, these mice, this is a picture basically, again, comparing sort of normal blood cells versus leukemia. This is the spleen of a normal mouse with a lot of lymphocytes, the spleen of a leukemic animal filled with leukemic cells. What's shown here is that the CDKN2A null mice, which is something, this change is seen in about a third of children with leukemia, have an abnormality of CDKN2A. But when we add in the ETV6-RUNX1, tel-AML1, they get sick more quickly, and they develop, half the mice develop B-lymphoblastic leukemia. So we've done very preliminary work looking at environmental exposures. We started out basically exposing the mice to radiation, and then we looked initially just at 5 weeks of age to see was there an effect of the radiation exposure on the development of the cells. Has it increased the immature B cells that express this protein? And does the exposure accelerate disease? And what we saw, in fact, that if you look, let's say, at the immature B cells, that the irradiated mice, a much greater proportion of them now express this ETV6 RUNX1 fusion. And what might be happening is the presence of this protein, basically what it does is that if there's something harmful that may be mutagenic that the animals are exposed to, the cells that sort of survive that and grow following that exposure are the cells that express this fusion. These are some data basically following mice over time that have gotten exposed to radiation. These are the CDKN2A null mice on their own. These are when the TEL-AML1 is present. There's a very small change, not reaching any kind of statistical significance when we irradiate the normal mice. But interestingly, there is a significant change of mice that develop leukemia early after radiation. It's almost as though there's a group of animals that when they get irradiated at a relatively modest dose, that they'll go on and they'll develop leukemia quickly. And then if they don't develop leukemia quickly, they develop leukemia just like every other mouse. So that the radiation either accelerates a subset or the mice are not affected by it. And so this is some evidence that we can use this model to look at the effect of exposures. So strengths of our model as a mouse bioassay include that it does model genetics of human pediatric BALL. We get the disease, pretty frequent disease. You'll notice the latency here is it's reasonably long, like 4 or 5 months, which is actually pretty good because it gives us some room to speed things up on exposure, and we don't have to wait for 2 years to get results. We know that radiation exposure can increase the frequency and decrease the penetrance of BALL, and we can use it. Limitations, it uses 2 genetic events rather than 1 or none, and one of these events isn't actually known to occur prenatally. All the mice get sick. So if we're trying to distinguish, getting ill versus not getting ill, it's not the perfect model for that, and it's genetically homogeneous. So this is a summary slide. It'll show up in the webinar that I think this is all going to be posted. But here are some potentially useful models that are available for modeling environmental exposures. And so these are the things that we're working on currently, using it in these ways. And in terms of the early endpoints, we're looking at, is there a change when we do an exposure? Does it change the lymphocyte numbers in the animal? Does it change the cells that express this pre-leukemic lesion? Does it make the pre-leukemic cells gain an advantage after an exposure? And we can do these with relatively low numbers of mice. And then there's a technology that enables us to basically ask, is there a change in gene expression? If we look at every RNA, every basically messenger RNA, transcript encoding a protein in the B lymphocytes after an exposure, are there any changes? And this can be done for basically every gene. So and we can also ask, you've heard epigenetics mentioned today. One way of looking at epigenetics is methylation of the genome. We can look at the genome. Does an exposure change methylation of any of the genes in a statistically significant way? Now, of course, when you look at thousands of things, you have to be very careful to avoid false positive results. And again, we can do this with relatively small numbers of mice. I want to leave my last slide to acknowledge people who have contributed to the work. And I thank you.